So the calorie epigenetic data at baseline generated by this pilot study, broadly speaking, are intact. Okay. So what we'd like to know now is if we can use these data to estimate the rate of aging. And so Horvath has his technology, and we're going to use that. But we'd also like to do something else. We'd also like to know about the rate of aging as measured by prospective analysis of the same set of individuals followed over time in parallel to what we're trying to do in the calorie experiment, where we're following the same people from time 1 to time 2 to time 3. So we did a study a couple of years ago where we followed 1,000 individuals for 12 years, measured them at 3 time points, and we had 18 different measurements of the physiologies of these individuals. And in this study, we tried to estimate the rate of aging based on differential rates of change in these 18 measurements, which ranged from standard blood chemistries, cardiorespiratory fitness was in there, telomere length, lung function, gum recession, all things that are known to change with age and that, in fact, we observed to change with age over this 12-year interval. We quantified the rate of aging as how fast, let's say, person A's rate of change was compared to the sample average. So we have a normative rate of change across these 18 markers, and then we're calling fast aging a rate of aging that is faster than that norm. Slow aging is a rate of change that is slower than that norm. Those rates of change estimated from these physiologies in turn were correlated with the functioning of these people. In that study, measures of physical function, cognitive function, how old their faces looked, were all correlated with how fast their physiologies were changing. So we wanted to know if we could take that measurement and put it into calorie. And to do that, we're going to use the same approach that Horvath and his colleagues have used to produce epigenetic clocks based on the chronological age of tissue samples. The technology is called elastic net regression. It turns out it's actually a fairly straightforward machine learning regression technique. And I'd be happy to talk more about it with those of you who are interested. We can produce a rough estimate of the pace of aging of these individuals based on a single time point of their methylation data. So we're sampling their methylome when they're 38 years old, and then we're looking backward at their rate of change over the preceding 12 years. So that's what this prediction equation is doing. And based on a cross-fold validation analysis where we train the algorithm in 90% of the sample, predict into 10% of the sample, and then repeat that 100 times, we can get a distribution of what the expected correlations between this measure and the actual observed rate of change in the physiologies is. It's not awesome. It's about 0.25. So we certainly can't recover the pace of aging as well as we could recover the chronological age of individuals in a mixed age data set, but there's some signal there. And this is what the distribution of values, when we take this algorithm and apply it to the calorie methylomes, looks like. It looks almost exactly like the distribution in the original cohort, with some individuals predicted to have essentially the same physiology at the time of the methylation collection as they had a decade previous, and others whose physiology is predicted to have changed at twice that rate. So that's what the scale is from 0 to 2 there. A value of 1 would indicate a rate of physiological change consistent with the chronological time elapsed. Okay. Sorry, those are just comparison prediction equations. So we wanted to know if the elastic net regression was doing a decent job of measuring the pace of aging. It's a little bit harder to predict the pace of aging, which is a complicated measurement than a simple hematologic parameter, so in this case the lymphocyte count, but we're doing about as well as we can for predicting something a little bit more complicated like BMI. I thought I had cut this because I actually think it's kind of complicated, but if you have questions, please ask. This is the distribution of expected correlations out of sample. Yeah, so that's a... This is a slightly more persuasive validation, I think. Bill mentioned the kind of phenotypic data that have been generated in CALERIE, and these are very much aligned with measures we studied in the original cohort. But there are tests of strength, grip strength, and knee flexion. There's gas exchange, cardiorespiratory fitness from a treadmill, and then there's the Cambridge battery for neuropsych assessment. And we would expect that people who are aging faster should be physically weaker, should have poor cardiorespiratory fitness, and should have poor neuropsychological functioning. And that's broadly what we see. So the data for the pace of aging algorithm are in light blue here. There's the correlation. the scatter plots are bin scatter plots, so because the graphs are a little cramped. So this is the average x and y coordinates for 5 individuals for each dot. The regression line and the correlation are computed from the raw data, though. So you can think about these broadly as confidence intervals. They show you the distribution of values in the data set. The dark blue dots are the sort of favored measure of epigenetic clock for this kind of prediction. It's what Steve and his colleagues are calling these days extrinsic epigenetic aging. And you see, as predicted, people who are aging faster by the epigenetic clock measure, by our measure, are physically weaker. With respect to cardiorespiratory fitness, we don't see an association with the epigenetic clock measure, but we do see this association with the pace of aging predictor. Yes?
Are you controlling for age in these analyses, or because it's a lot easier just to measure chronologic age?
That's right.
So many of these models look for sort of the deviation of your chronologic age.
Yeah, so the question for those of you online was, are these associations adjusted for age? And in fact, I think these are not. If you adjusted for age, you'd get almost exactly the same thing, though, because this epigenetic age acceleration measure is based on a residualization of an epigenetic clock for the chronological age of the tissue. So essentially, the predictor variable here is already controlled. And in this case, everybody who was involved in the training of the pace of aging algorithm was the same age. They were all 38 years old. So the only content in this epigenetic prediction algorithm is variation in the observed rate of physiological change in those individuals. Yes.
Dan, you keep saying tissue, and you know there's an age-related drift in population heterogeneity, and Steve for blood has an algorithm to compensate for the fact that you're having fewer naive T cells, more memory T cells, so on. So you want to elaborate on that aspect of the correction for what tissue you're talking about exactly, and how you deal with shifts in population heterogeneity?
Sure. So the answer, I think very briefly, is that for this analysis, we don't. So we're accepting that the composition of blood changes as people age. And from the perspective of these algorithms, that's signal, not noise. So if we want to know how fast a person is aging, how the composition of their blood is changing is potentially information. We could certainly adjust these analyses for the leukocyte composition of blood samples. It's going to make a difference in this case, because this extrinsic epigenetic aging turns out to be, in fact, quite correlated with specific lymphocyte populations that are known to change with age. That's not the case for either the physiologically measured pace of aging or the epigenetic prediction of the pace of aging. But we can certainly do some sensitivity analyses to see, with the information we have, to what extent these correlations are dependent on the composition of the blood sample. At least in the preliminary looking that we've done of the cell counts estimated by Hausman and the calorie complete blood count data, this predictor isn't giving us very much information about what the cell mix of the blood is. So I would expect that not to change very much. But it is the case that we're dealing with whole blood here. These are the cognitive data. This is a sort of a surprising result, but I think, you know, it's a small sample, so we can write that off as 0, I guess. Essentially, the regression line is going the wrong way. So people who are epigenetically aging faster, according to this clock measure, have superior cognitive function, which is a bit of a surprise. This is what we would expect, which is that faster aging is correlated with poorer cognitive function. So that's not the really interesting question, though. The interesting question is, does the epigenetic predictor at baseline forecast the subsequent rate of aging in calorie? If we assume that these are both essentially looking backward at the history of an organism and saying, this is roughly the aging rate as of today, that's more or less the prediction of the aging rate for tomorrow. And what we'd like to know is, does that prediction hold? Can we forecast the rate of aging in this organism over the next 2 years during calorie? And is that rate of aging altered by the intervention, by caloric restriction? So this is the answer for our predictor for the pace of aging. This is the ad libitum group. And these dots, now I've got 1 dot for everybody because I've got plenty of space on the figure here. And what you can see is that for those individuals who were not subjected to the caloric restriction intervention, the prediction of their aging rate, as measured by the change in that biological age that we were using before, is basically what we expected it to be based on our cross-validation analysis in Dunedin. So people who have an epigenetic prediction of a faster rate of aging are aging more rapidly in the ad libitum group in CALERIE, and the effect size is about 0.25. In contrast, in the caloric restriction condition, there's essentially no association between your epigenetically predicted rate of aging and the rate of change in your physiology that we observe over CALERIE, parallel to the other analysis that we performed. So, again, this is a very much incomplete project, and these data only arrived, again, last week. So we know a little bit, we'll know a lot more in the coming weeks and months, and there'll be a paper soon and a data set for those of you who are interested. And in the meantime, if you're especially eager, you can contact me. Here's a few other odds and ends. So this is that same figure drawn for the extrinsic epigenetic age acceleration predictor. Again, sort of a puzzling result. Faster epigenetic aging predicting slower rate of change. And here's intrinsic epigenetic aging, and this is a completely cell mix independent measure of biological age based on blood DNA methylation, or it's the best that Steve has been able to approximate so far. And again, it's not what we would expect in these data. I should say the epigenetic clocks were not built to serve as surrogate endpoints in geroprotector trials, and that's not the research question their inventors are primarily interested in pursuing. So I don't know that these data tell us anything about those clocks per se, although they may encourage caution in moving forward with using them in this surrogate endpoint context. That said, we'd very much like to find out how these epigenetic measurements change over time in CALERIE, and with that in mind, we hope to raise funds to assay the methylomes at the subsequent time point so we can in fact compute slopes of epigenetic aging.